Good afternoon, everyone. Thank you for sticking around. Today, my talk will focus on the influence of host genetics on HIV-1 acquisition and vaccine efficacy in the RV144 study. So the RV144 HIV-1 vaccine trial conducted in Thailand showed an efficacy of 31%, and this was comparing placebo-infected cases to vaccinated infected cases. A follow-on study looking at immune correlates of protection comparing vaccinated infected cases to vaccinated uninfected controls identified an HIV-specific antibody that correlated with decreased risk of acquisition. And this was IgG binding to a region of the HIV-1 envelope, and it correlated with decreased risk of acquisition, so it was protective. And this antigen is on GP70 amino acid residues 120 to 204, and it consists mainly of the variable V1, V2 sequences and some conserved C1 and C2 sequences on either ends. So high IgG responses to this antigen was protective in the RV144 study. I will refer to IgG responses to this antigen again as IgG in the rest of my talk. So we see that variation in IgG responses to this antigen were protective. So since there is this variation among individuals after vaccination, we asked if this variation could potentially be due to differences or variation in the host genes, particularly HLA class II genes. And this is because HLA class II genes encode for HLA class II molecules, which are expressed on antigen-presenting cells, and they present peptides to CD4 T cells, which then stimulate B cells to produce antibodies. And this is why we looked at HLA class II genes. So today I'm going to talk about the HLA class II associations with acquisition and efficacy. And all these, what I'm going to present today has been published in the Science Translational Medicine Journal last week. So coming to the study design, we asked two main questions. Do HLA class II genes correlate with IgG antibody levels and affect acquisition? And two, do any of these interactions have an effect on vaccine efficacy? And so we used the same study design as was used previously. And we genotyped individuals for classical HLA class II genes, DPB1, DQB1, and DRB1, and we tested the association of 31 class II alleles. And of the 31 alleles, there was only one allele that associated with increased levels of IgG. So here on the y-axis, we have IgG antibody levels, and individuals that were DPB13 positive had higher levels of IgG antibodies compared to individuals that did not have DPB13. So what about its effect on acquisition? So we stratified the same data by infection status. And here again on the y-axis is IgG antibody levels. And here, in addition to stratifying by DPB13 presence or absence, we also stratify by infection status. So in the absence of DPB13, there is no difference in IgG levels comparing infected to uninfected individuals. But in the presence of DPB13, we see that individuals that were uninfected had higher levels of IgG compared to individuals that were infected, showing that high IgG levels are a correlate of decreased risk of HIV-1 acquisition only in the presence of this single HLA allele. So what about its effect on vaccine efficacy? For this, we categorized IgG antibody levels into high, medium, and low, and also stratified individuals by presence or absence of DPB13 and estimated vaccine efficacy. And of the 4 groups, individuals that had high, medium IgG and DPB13 positive, that group was the only one that showed efficacy, and the efficacy was 71%. So here what we show is that individuals that have both high IgG levels and this particular allele have decreased risk of acquisition and increased vaccine efficacy. So in order to understand this mechanism better, we looked at IgG responses to 181 overlapping peptides spanning the on antigen, and we asked if any of them are more common in DPB13 positive individuals compared to DPB13 negative individuals, and we identified 5 epitopes. So IgG responses to these epitopes were more common in DPB13 positive people than negative individuals. And of these 5 responses, one epitope, IgG responses to this epitope was more frequent among vaccinated uninfected individuals compared to vaccinated infected individuals that were, again, DPB13 positive. And we see the same thing with magnitude of IgG responses to this particular epitope, 119 and 133. So if we look only among DPB13 positive individuals, individuals that were uninfected had higher magnitude of IgG responses to this epitope compared to infected individuals. So what we're showing here is that magnitude of IgG responses to on-peptide 119 to 133 confers protection in DPB13 positive individuals, and this is the same with frequency of IgG responses to this peptide. And this peptide is important because it binds to CD4, and also this region is very conserved among different viral subtypes. So here, what I've showed till now is that individuals with DPB13 are having vaccine-induced responses that are protective. So could this differences in immune responses have an effect on the virus? So for this, Morgan Roland from MHRP looked at viral sequences of breakthrough infections and looked at particular sites which have known function. Either they were antibody epitopes or they were contact residues. And she stratified viral sequences by presence or absence of this DPB1 13 allele. And among the placebos, she did not see any differences in viral sequences comparing 13 positive to 13 negative individuals. But among the vaccinees, there was one position, 173, where histidine at that position was more common in DPB13 positive individuals compared to the negative individuals. So what I'm showing here clearly suggests that there is some difference in immune responses in DPB13 individuals after vaccination. So to summarize the findings, presence of HLA DPB13 correlates with higher quantities of IgG antibody levels and associates with decreased risk of HIV-1 acquisition and increased vaccine efficacy. We also see that both frequency and magnitude of IgG responses to a particular peptide, 119 to 133, confers protection in DPB13 positive individuals. And vaccine-induced responses are exerting pressure on the virus in individuals with this allele. So what we see here is that HLA class II genes are modulating the quantity of antibody responses and efficacy observed in the RV144 study. And differences in vaccine-induced responses elicited by these individuals with this particular allele need to be examined closely, and understanding how variation in the host relates to the vaccine-induced responses clarifies interpretation of vaccine efficacy studies and could prospectively improve HIV vaccine design. I'd like to thank all the people involved in this work. This would not have been possible without the contribution of many individuals at MHRP and individuals at other institutes. I'd like to especially thank Heather Prentice, a former postdoctoral fellow in my lab who did a lot of the statistical analysis. Thank you.